Uh, so in our department now since 2011, so almost a decade. It'll be a decade in July. And I haven't actually taught any undergraduate classes for a couple of years, in large part because we've been developing this master's program, and that's been my focus for the last 3 years now. But many of you, if you're seniors, I will see you in the winter for AIChE 118, Process Dynamics and Control. That was a course that I taught for my first 6 years, and I'm happy to start to teach that again. So we'll see you in the winter if you're in that class. But today I wanted to talk about our new Center for Industrial Biotechnology and more specifically about the master's program that's associated with this. We're now in our 3rd year. There's a 3rd class of students who are in this program. And then I'll tell you about biotechnology or industrial biotechnology and the program. So this is it in a snapshot. It's a professional science master's, or actually it's a degree in chemical and biological, or sorry, not biological, we're not a biological department, we're a chemical and environmental engineering department. And so that's actually the name of the degree, but this is a specific track on industrial biotechnology. And so what that means is it's a master's program that you can finish in as minimum, as low as 9 months, or as maybe up to 12 months, depending on when you take the internship part of the course. There's only a few programs, I think, like this across the country. There's maybe one in Berkeley has something, but it's significantly different and it's kind of directed towards product design. There might be one at Northwestern up in Chicago, and then maybe a couple others that I haven't listed out all the details of yet. And so certainly in our area, we're one of the only programs that are like this. The other would be our friends at the Keck Institute, Graduate Institute, which has a 2 years master's program that's a little bit more involved than ours. So really, we're focusing on 4 different areas, sorry, and I'll talk about those in a few minutes. But industrial biotechnology is a pretty big sector in the United States, and I think this is a program that will prepare you to move directly into that industry. There's lots of different faculty who interact with this program, both in the master's program side of it as well as the research side, and you know, we'll get into the details now. Industrial biotechnology, if you don't know, it kind of addresses these 4 different topics, or this is how we think of it, and addressing things like healthcare and medicines, food and pharmaceuticals, certainly biofuels production and industrial chemicals production, and all of these certainly have their benefits, right? In the case of health and healthcare and medicines, you can see that on a daily basis right now in terms of vaccine development or in terms of testing for COVID. Right. If you look at, or if you've been following the news about the vaccine development at Pfizer and now Moderna, the next issue for them is going to be scale up. And that's certainly a topic that we intimately address in this, 9 to 12 months that you'd be involved with this program. Really, that's the focus of the program, which is how do you design a bioprocessing plant, right? And what are the skills you need to do that, and then what are the softwares and other fundamental skills that you need to be able to accurately design a plant. And so certainly making products for the healthcare industry, or making products for the food and agricultural industry, making biofuels, and then also making industrial chemicals. These are big areas within industrial biotechnology. If you look at it across the country, this is a pretty big sector of the economy. In the US, it's about 2 to 2.5% of GDP. And this is an industry that's been growing at a rate of 10% per year over the last decade. So we get about $400 billion in revenue, and this has been going up again every year. If you look at just the if you look at that total amount of money in there, this is revenues from that industry. It's about one third to GM crops, the production of biologics, right? So that's things like the vaccines that we've been hearing about in the news, or industrial chemicals. I show here the industrial biotechnology revenues, the industrial chemical revenues here, because this is a focus of my own research group. And that in how do you, produce different chemicals that would go directly into the chemical kind of feedstock industry. Those things can include, of course, biofuels, additives for food and agriculture, biological feedstocks, so the inputs to making different biologics, as well as just biochemicals overall. So there's a report that I've seen recently, and Sorry, here it is. There's a report that I've seen recently, and this has been updated, I think, every year for the last little while. And so here I'm showing the 2020 data. This is a report called the California Economic Impact Report for the Bioeconomy. This tells us, or this analysis shows us that there's about 1.4 million jobs across the state, about $4.5 billion in research funding from the NIH, that's the National Institutes of Health, that come to California. And there's about over 15,000 different establishments. So that means different businesses or companies that operate in California that are directly related to this industry. This is a pretty substantial report, and we can get into a little bit more detail of it. And certainly one of the parts that's interesting, I think, to most people here is how much money different parts of this sector, what the average wages or average salaries are in different sectors of this industry. So this includes a lot of topics here. Not all are directly addressable by this master's program, but certainly biorenewables, the manufacturing of biopharmaceuticals, diagnostic devices and medical equipment. Certainly that's something that you could think about here. Life science wholesale and research and lab services as well. Those are all the lab services part, maybe not, but most of these kind of topics here are directly addressed in our program. We really would focus on the production of biorenewables or biomanufacturing for pharmaceuticals. Those are really the two main areas that we focus on. There are considerable wages, I think, here. And if you dig a little bit deeper into this report, it'll start to tell you things like what is the average educational attainment of this sector as well. And so, yeah, if you look at the biorenewables, this is an average low wage, but this is where I think most of the individuals that have gone through the program have gone into this sector. And certainly they're beginning at salaries that are much higher than this. They're closer to the $80,000 or $90,000 range. And I think that's because they have this higher educational attainment to begin into this, or to get into this industry. And this report also gets in a lot more detail, and you can find it yourself. I think in a few slides ago, this slide, both of these slides, it's at this website here, biocom.org. You'll find the full report as well as a breakdown of different areas. And so this is a local snapshot that I wanted to share about Southern California. And so we can see here, it's got Riverside County in here, and it kind of mirrors the statewide averages. Pharmaceutical manufacturing is a little bit high, medical devices is a bit higher than that across the country and across the state. Probably biopharmaceutical manufacturing is maybe the highest average wage. But this gives you a good idea of what jobs are available. And again, if we go back to that idea or that state, that stat a few slides ago, which showed that there was over 15,000 different entities or companies that are in this industry across the state. That's a large number of opportunities I think that are available for chemical engineers to move on to, particularly if you finish this program, which would make you, I think, a certified biochemical engineer. So this is the program. This is the curriculum for the program. There's 3 quarters plus some work in the summer. That summer work, and we can start there, is about an internship. And so there's a minimum requirement of 2 units, which is about a 6 to 8 week internship that we'd hope you do in industry. It's also possible that you do that on campus in a different research lab, or one of a number of different research labs. And so last year I had 3 of the students in the program do their research internships with me. Other labs are certainly available as well. But that's the end of the program, right? And so we'd like you to finish off with getting an opportunity in industry or in a research lab on campus or at a different campus to learn some practical hands-on skills. Other examples of internships have been at Illumina, the DNA sequencing company, also at IPS, which is a design company in a bioprocess design company in Irvine, a small startup company as well. Needed some design work, and an intern, somebody at the program last year went to do an internship with them. So that's the end. You begin in the fall quarter with taking Chemical Engineering 124 and 124L. This year 124L, because of COVID, has been pushed off to a later spring quarter, but we hope to get back to this kind of curriculum and plan next year. So 124 and 124L overlap with of course, they're the undergraduate courses that you might be taking as a senior in the biochemical engineering option. So if you've taken those already, then there's other courses that we can fill in there if you move on to this master's program. So 124, 124L, that's part of the fall. Dr. Wyman has a course called Energy Production Uses Economics and Sustainability. This is part of the fall quarter as well. That's, I think, a very useful course to look at generally overall how energy flows through these different systems. And in this case, it's related to biofuels, or the topic that's most interesting to this program is the biofuels aspect of Dr. Wyman's class. And then you also start the first of 3 classes that are about bioprocess design. And so they're here all listed as CEE 238A, B, and C, Bioprocess Design Laboratory 1, 2, and 3. And so this to me is kind of the core of the program, where you start to learn more detailed aspects of the software called Super Pro Designer. And that might be a software that you also get exposed to in your senior design project. And when we really like this software for bioprocessing, because there's a number of units and elements of that software that are directly related to bioprocessing. The software was made by a gentleman, Dimitri, who we know reasonably well in the program, and actually a number of us attended one of their seminars last year, and he's been very gracious in letting us use that full versions of that software that we can all have access to in this program. And so there's a lot of inherent bioprocessing aspects of bioprocessing built into the software. And so there's things like specific fermentation units or specific types of fermenters that would be applicable to, say, antibody manufacturing that are already built into that system. And so this is a design software where you can start to look at the process overall and design different processes, and then start to think about scheduling of that process over the course of a year. How many runs through the plant can you do? When is that different piece of equipment going to be occupied, and so do some of those optimizations as well. So in the first quarter, you start to really look at upstream processes. So that's the fermentation part. In the second quarter, we look at separations in terms of process design. And then in the spring quarter, you kind of look at the overall process and start to build up full processes where you've learned the upstream and downstream skills in the fall and winter quarters. Of course, as a graduate student in the department, you also take CE 286, which is our seminar. This is the Chemical and Environmental Engineering Seminar, where every Friday morning we have an outside speaker come, now via Zoom or in the past, and hopefully again in the future, come in person to give a lecture on their topic of choice, right, of their topic of research. And often we get some really good biotechnology or industrial biotechnology speakers. A number of years ago, maybe only 2, maybe 3 years ago, we had Frances Arnold come. And so Frances, of course, won the Nobel Prize just this year, or last year, excuse me, for her work in directed evolution. So that's in enzyme and protein engineering. So in the winter quarter, we're going to have bioseparations. That's really an advancement on what you learn as a senior undergraduate in separations, but it's of course dedicated to bioprocesses. Then you also begin some more specific information about upstream processes in biotechnology. And so what we mean by upstream is that's all the things before separations, right? How do you grow cells? How do you start from a really small flask of cells, or how do you start from the freezer stock of cells, grow a little bit of cells, expand that culture, and continue on until you get to a 1 million liter reactor, right? And so how do you do those designs? How do you start to implement them in Super Pro? Of course, that's in Bioprocess Design Laboratory. But in the winter quarter, you start to really advance on your calculations that you would have begun in 124 and start to learn about some of the more advanced elements of upstream processing. That also gets into the genetic engineering of the different microbial and mammalian cell strains that you might be using, right, for your production. So again, then you also have bioprocess designs laboratory 2, and so you focus on bioseparations in the winter, and again doing your seminar as well. In the spring quarter, you have cell engineering. This overlaps with CEE or CHE 140, which is also called cell engineering. You're taking now the graduate version of that class. Again, if you were a member of the biochemical engineering option in the undergraduate, you would have taken this class already, and so there's a number of other options that you can take in place of that. You follow along with C248. That's additional calculations for the upstream part. Anything that we missed in the first 2 quarters, then you start to fill in this last quarter. And then, of course, you take a bioprocess design laboratory 3 in the seminar as well. So really, what I think you get here is a really dedicated a set of courses on one topic. And so I think you can come into this program with not knowing very much about bioprocessing at all, or much about biology or biochemistry or biochemical engineering overall, and you'll have 9 months of dedicated work here, right? And you have the courses then that will fill in maybe the background that you didn't have before, if you hadn't been in the biochemical engineering option, or if you haven't taken biology since high school. I guess you probably all have to take Bio 5A, and so there'd be rare exceptions that you haven't taken biology since high school. But if you've only had just a minimum of Bio 5A, you're still a good applicant, and I think a good student in this program. You're going to dedicate yourself to this, learning this topic for 9 months, and then also do an internship, so it could extend it to 12 months. That's a lot of time to really learn a focused set of skills. And I think that's the value of this type of master's program, where as opposed to maybe if you're thinking about a BS MS program in our department, if you do the MS side of it, it's just going to be some extra things that you didn't pick up in undergraduate. So you'll do kinetics again, you'll do thermodynamics again, you'll do transport phenomena again, you'll learn some new calculations, but really you're just kind of enhancing your chemical engineering skills that you had already developed. In this case, for that 1 year afterwards, that one academic year afterwards, and the MS to follow along with your BS, you're really learning a dedicated set of new skills. And I think all of these courses together start to, give you the ability to translate yourself directly into the industry. So the next few slides I have, please stop me if there's any questions. This is certainly not a formal presentation, and I have not really been following if there are hands up in the participation or anything, but in the participation window. Sorry. Okay, so the next thing that I wanted to show you is just some of the examples of things that you'd be working on in the bioprocess design laboratory. Oh, there's something in the chat. Here it is. Oh, please sign in. Okay, I won't sign in. So one of the things that, and I kind of moved these examples up first because we're talking about it in the media so much these days, which is antibody production, or maybe more importantly, vaccine production. And so one of the classic examples of bioprocessing is, the production of antibodies, right? And so we saw when the president got sick from COVID a few months ago, that one of the treatments that he was almost immediately given was this antibody bolus, right? And those are antibodies raised against the SARS-CoV proteins, right? And so specifically, those are going to be raised against, which means they are designed to bind to the infective parts of the virus. And so on SARS-CoV-2, it's a spike protein that comes out of the virus, and on top of that spike protein is this receptor binding domain. And so this is a piece of that protein on the outside of the virus that binds to your cells, and that's how the infection begins. One of the ways to treat against, or to treat COVID, is to have a different protein bind to that receptor domain, and therefore block its function so that it doesn't bind to your cells and then get inside your cells and then replicate, etc. And so one of the things that you have to do then is to find those antibodies. And so that's antibody discovery. And if you've talked to Dr. Ge in our department ever, that's one of his major focuses of his research group. Is that the next step then, once you have that antibody, or if you identified an antibody that you think can do the function that you're looking for, in this case, binding to that receptor binding domain, or RBD of SARS-CoV-2, you have to make a huge amount of it, right? And so designing that process, and then, designing that process like this one here, and then building that process is certainly the next step. And so this is a good example of a classic bioprocess here, where you've got a seed train, which is generation of the cells that you need for the process. So you start from the inoculum, or you start from the freezer, right? You take a stock of those cells that you need. You start in a small flask called a T flask, and then you grow those cells up so it's to confluency, meaning you've got a full culture there. You translate that culture, part of that culture, into a bigger flask and a bigger flask and a bigger flask, all the way to you get to the right production scale that you're looking for. And so now you move into this production phase of the process overall. You do your reaction, your fermentation, or your long, 7 or 12 day or some multi-day process in here where you're producing your product. And then you stop that product, and then the rest of the bioprocess is about removing everything else and just trying to separate your product from from the rest of the cells and the other proteins that are there. And so those separations and purifications, certainly, you could have seen that in the curriculum that we had in the winter courses, the bioseparations, then of course the corresponding bio process design laboratory that should focus on that, but the separations design as well. And so really you can see here that separations and purifications in this particular process, and in many bioprocesses overall, dominate the process, right, or dominate the amount of work and units that are involved here. And just as in real estate, you've probably heard location matters. And here in bioprocessing, often separations matter, right? And so it's things like antibody separations is an enormous part of the cost overall here of making your product. So that's one example of things that you'll learn about in the bioprocess design class, as well as in the upstream and downstream parts of the course. The other thing I wanted to show you is just, a slide that I used as a lecture last year when I was doing this course in March, and we, quickly had stay-at-home orders, and we had to move to online teaching right away. And so we didn't have a lot of contact time with each other, and we kind of slowed down a little bit on our bioprocess design. And because this was happening, in real time for us, we started to look at the CDC guidelines for the SARS-CoV-2 RNA test. Right? And we started to analyze that protocol to see if we could find out parts of that protocol that might be causing delays in its manufacturing. And so in doing so, we learned how the test works and what the major parts of the test are, and then also started to look at new tests or new types of tests that people were coming up with very quickly. So I'm showing here on the left is the standard CDC test for individual PCR tests and how it works. But then on the right, I'm showing this multiplex screening strategy. And so this was something that a new startup company that I know of in the Bay Area had quickly pivoted their whole, biological workflow and research pipeline towards making a new type of test that you could maybe analyze up to about 10,000 individuals in a given day without a lot of automation. So the core here of the technique is DNA sequencing. And so Illumina is a company I referred to earlier. Illumina is a company that makes DNA sequencers, right? And so these are sequencers that can read 400 million base pairs in a few days' time, or more in some of their bigger machines now, or less than, of course, in some of their smaller machines. And so what this does is take a pool of DNA where you've barcoded each of those pieces of DNA so you know where they came from. And then they get put down onto like a silicon chip, and then you synthesize a strand of that DNA that's a copy of the strand of DNA that you've put on the surface, and you sequence by synthesizing, right? And so you synthesize, you add an A, and then you can read, oh, an A was added here, or you added T, etc. And so that's called sequencing by synthesis. And so what happened here, what this research group, this startup company came up with, was a strategy to extract RNA directly from samples from the swab, and they've called this method swab-seq. And so to amplify the RNA into DNA directly, and then be able to pool those into one big pool and read them on these sequencers. And so in doing so, by the kind of processes that are outlined here, they could theoretically then test about 10,000 people a day in one laboratory with only kind of manual labor and then sequencing at the end. And so this was interesting at the time to really look at how SARS, COVID-2 testing was happening at the time, what potential bottlenecks in terms of production were there, and then also what new tests were coming available. We spent a couple of weeks talking about this directly, and I thought it was really interesting. The other things that we do overall, or at least how I have approached this bioprocess design class, is to approach it in a way that we're, in most of the assignments, we treat ourselves as a small company and say, okay, well, let's do some, exploratory work in terms of looking at the patent literature to find, is there a novel process that we can identify in the patent literature that might be suitable for a small company or a big company for that matter? And then start to do some modeling around it to see if we can do some initial back to the envelope calculations to see if we think it's a viable process. Use the patents to get into some of the details of how we might expand that process into a bigger plant, and then start to do the evaluations of that plant overall. And so in this case, this is an example born out of some of the research that is done in my group, is the production of long chain dicarboxylic acids. And so if you think about nylon 6,6, right, the 6,6 parts refer to two precursors, each of 6 carbon chains, 6 carbons in length. And so adipic acid is a 6 carbon molecule that is a diacid. So there's an acid group on either side. That's the adipic acid. What we're talking about here is now moving, instead of those 6 carbon chains, to longer and longer carbon chains, which would allow polymers manufacturers and material scientists to really access different sets of polymers now. And you can do this quite well in biology, but you can't do it very well in chemical synthesis. And so what's something that biology is very good at is doing specific reactions through enzyme catalysis. And so what is possible through different enzyme reactions is to do these terminal hydroxylations that are shown here on the bottom, which is a P450 enzyme will specifically add a group to the tail of one of these molecules. Or either of the tails in the case of the bottom example. And this is a pretty hard reaction to do through chemical synthesis, because you get a real lack of specificity, meaning you'll get that carboxylic acid group added to the middle of the chain as opposed to the end of it. And so this is one way where biology can really shine, which is to do these specific and reactions that are not possible through chemical synthesis, or are very challenging through chemical synthesis. And so what we did here in this example was to say, look at a couple of different patents. There's 2 of them here. One is about purification of the products, and the other is about making these long chain dicarboxylic acids. And so we took these patents and the detailed information in them, and then started to build out processes, and then start to apply, is this going to be economically feasible or not? Or ask different questions about it. What's the size of the reactor you need to make X amount of stuff? How long do you have to do this fermentation for? Or in terms of separations, what's the best separations methods to get these long chain dicarboxylic acids out of the fermenter and into a purified form? And so we really like to try to build these around specific examples, the projects around specific examples, and say, okay, at the outset, here's just a 1-page problem statement. Our company is interested in developing this process. Let's explore it, and put our a little bit of acting on to say, let's act like we're this small company and start to evaluate if this is going to be a useful process or not, and ask some really pointed questions about those processes and about those patents. And so another type of example would be one here. This is again kind of inspired by research in my own group, and in this case we're looking at high temperature yeast. And so well, maybe you don't know, but it's pretty common that all of the ethanol that's produced in the United States and more generally across the world is produced through fermentation. And so you take this yeast called Saccharomyces cerevisiae, baker's yeast, and produces actually quite a bit of ethanol if you give it a lot of sugar. And so typically then you would take corn sugar that you grow in a farmer's field, you leave the plant and take the corn, you process that a little bit in a couple of different ways, and you extract glucose from that. And then in a large fermentation plant, you turn that glucose sugar into ethanol, right? And so my point here is that we're doing that with Saccharomyces cerevisiae, and that has to occur at a temperature of about 30 or around 30 degrees Celsius. What you might want to do is look at increasing the temperature to 35 or 40 or 50 degrees C and see if there's benefits at those higher temperatures. And so one thing that you would need then is a yeast that can tolerate those higher temperatures. And in this case, we have found one, and we work on one of them in my lab called Cluvariumyces marxianus, or K. marxianus. This is a yeast that, compared to Saccharomyces, grows at very high temperatures. And so Saccharomyces grows at 30, maybe 35 if you push it. And so K. marxianus can grow up to about 45 or 48 degrees C. And it can still survive temperatures of about 50. It doesn't grow very well, but it can survive those temperatures. It also grows much, much quicker than Saccharomyces. It grows like a bacteria does. It grows almost as quickly as E. coli does, and can do it at these elevated temperatures at 37 and 40 degrees C. And so one of the processes that we started to look at then was to say, can we make ethyl acetate? And so ethyl acetate is just um, acetylation of ethanol. And so there's a specific chemical, biochemical route in K. marxianus that produces ethyl acetate. And this is a natural product that's made from this yeast. And so we can ask the question now, with the skills that you'd have in this program, what's the effective temperature overall, right? And so we did this analysis, and we saw that, look, actually there's a big effective temperature, but it's limited to a switch between 30 and 35 degrees C. And so we know that, this is actually borne out in conversations with industry, that we know that, in industry, you want to move to a 35 or above, 35 C or above process. And that's at minimum, because you can now use a different type of cooling apparatus. At temperatures at 30 degrees C, you have to actively cool your fermenter, right? Or actually at all these temperatures, you have to actively cool the fermenter. But at 30 degrees C, you need chilled water, right? So you have to refrigerate your water to cool down your reactor. And the reactor heats up because a lot of the energy in the glucose is released as heat, right? And so you have this growth of the bacteria, or the yeast in this case, in the fermenter, and that starts to heat up. And so to maintain it at 30 degrees C, you need active cooling, right? To maintain it at 30 degrees C, you need cooled water, or water chilled and refrigerated down to 15 or 20 degrees Celsius. At a temperature of 35 degrees C, in most climates, you can get away with a cooling tower, meaning you pump the water up to the top of the tower, and then you let it drop down, and it cools by evaporative cooling. And so this key switch here, in terms of a piece of equipment, is identified by SuperPro as one of the crucial parts here in terms of increasing temperature and the cost of savings that you can get there, right? So if you go from 30 to 35 degrees, you're changing the type of cooling water you need for the fermenter, and in doing so, you can save about 8 times or 8-fold in the normalized utilities cost. And so really, you're saving a lot of utilities cost by just making that one switch in a piece of equipment, right? You also have confounding effects here in terms of how quickly the reactor will go, or how quickly your fermentation will go. And so you can see, if you start to model the reaction kinetics or the growth kinetics of in this case the yeast, you get faster and faster growth in a smaller and smaller batch time at higher temperatures. But of course, it plateaus after a while, and it also would go up considerably if you get too high. And so, these conversations, or this conclusion here, that moving from 30 to 35 degrees is an important aspect, is borne out by our conversations with people in industry. And so this is also, I think, a really nice example of how you can use SuperPro and kind of operate in our design class to ask real questions about bioprocessing. So not only related to the center is there this master's program, but there's also active research going on. And these are places that you can think about doing research for your internship, or just interacting with those faculty as well. Again, the themes of our research are mimicked overall by the themes of industrial biotechnology, production of health care and products and medicines, food and agriculture, biofuels, and industrial chemicals as well. The people involved here are, of course, myself. I'm the director of the Center for Industrial Biotechnology, and also spearhead the master's program. Dr. Harvey Blanche, he's part of our faculty as well. He spent most of his career at Berkeley and retired a few years ago, more than a few years ago now, and then has joined us part-time in our department. He's really been instrumental in helping us, get this center and the master's program off the ground. Dr. Guh, you may have had in separations this quarter for your seniors. Dr. Jinkerson joined us a few years ago, and so did Dr. Lee. I'm sorry, there's a typo on this slide. It's not Dr. Liu, it's Dr. Yanran Lee. I'm gonna fix that. They both joined us a couple of years ago and are, of course, intimate in this program. Their research is directed towards algal biofuels and algal research for Dr. Jinkerson, and then also how do you move plant biosynthetic pathways into yeast hosts. That's Dr. Lee's work. And then Dr. Weinman, of course, is his course I talked about. As also, he's been doing biofuels research for the last 35, 40 years. We also have our partners in botany and plant sciences, Dr. Bailey Sayers and Dr. Cutler and Dr. Nelson. We just had a kind of research retreat last weekend talking about a different training grant that's involved chemical engineers in botany and plant sciences researchers as well. So that's a very good interaction with us for them as well. Okay, so how to apply? This is where I'd like to end. Excuse me. If you're applying just the you apply to this program just via the typical MS program, but you indicate that you're interested in the industrial biotechnology track, right? So there's no special application. It's just the standard CE MS application. You can apply through the BSMS program as well. Really, this is not a unique degree. It is a specific track of our MS program in CEE. Right? So you get a degree in chemical and environmental engineering. The focus is, again, directly on industrial biotechnology, and the courses you take are not the same that you would take in a typical master's program in our department. It's those courses that I outlined in a few slides ago. Those are the only courses you have to take. The other part here that's a little bit different from a traditional master's program is that there's no test at the end, right? So in our typical master's program, there's a cumulative test at the end that will test you in thermodynamics, transport, kinetics, thermodynamics, transport, and kinetics. If you're a chemical engineer or if you're an environmental engineer, it would involve some air quality engineering and water quality engineering. That test is not required in our program, in the Industrial Biotechnology program. In place of that, you have to write a report about your internship. And so at the end of your internship, wherever it might be, whether it's on campus or in industry, you do have to write a report of what you did there and convey that information to us. That report is in place of taking a standardized test at the end of the year. And so the requirements for the BSMS program, I believe, have not changed. You should have a cumulative GPA of 3.4, minimum of 3.2 in math, science, and engineering courses. That's automatic acceptance. GRE scores, I believe, have been waived for all applicants this coming year due to kind of COVID restrictions on access to that test. We as a department are hoping, I think, to get rid of that as a requirement going forward, regardless of COVID or not. The link to how to apply is there, of course, and you can find it on the department webpage as well. The deadline for applications is actually September 1st, 2021. So you have a long time to think about this. And I'd like to, bring this up again and later in the year as well, if you would like to talk about it again. The traditional deadline for applications to the graduate school are coming up. So if you're looking at a PhD program, it's early December, or in other cases, in other schools, maybe early January. And so, of course, our application window is open now. If you're interested in this program, I suggest you apply as soon as you know that you're interested. But again, if you're still thinking about it up into the spring, you have until the end of summer to decide to apply. And of course, the timeline for applying, if you apply on September 1st, I'm not sure if we can turn it around fast enough to get you in by September 2nd. We would do our best, but I do suggest that you try to apply before this time, or at least that you notify myself and other people in the department once you've applied, if you've left it that late. Regardless, I'd also like to hear if you're applying to this program at any time. And so please contact me if you have any questions about the application process, about what we do in the program, or anything in general about our department, or about process control and dynamics, which I'll be teaching some of you in the winter. That's all I have. That's all I have to share with you, but I'm happy to answer any questions that you guys might have.
So you mentioned that the program's been around for about 3 years. Do
Yes.
you know what some of the graduates have been doing after they got the degree?
Sure. Yeah. So we're just in our third year now. The first year we only had 3 students. The second year we had 5, and then this year we have 3 again. And so, one, I just note that it's been pretty small classes, which are probably distinctly different from some of your classes in undergraduate now. I know that our process dynamics and control is almost at 80 for the winter quarter. So you'll be going from that to a much smaller class. And we do anticipate to grow a little bit, but I don't expect us to be much bigger than 10 or 12, maybe 15 students. Okay, so the more interesting answer here is yes, I know what they're all doing. 5 of the 6 domestic students, 2 of those students that I listed were international, and I actually don't know where they are now. I know they've just gone back home. I just don't know what they're doing. But 5 of those 6 students who have graduated with the program found a job within, I would say, 1 to 2 months after graduating. One is in my lab now. Still, I just hired him on as a researcher in my group. He's also helping to manage the lab. That's not because he didn't have other opportunities, but that's because he was continuing some work that he wanted. The first student, the first domestic student in the first year, she got an internship at Illumina, and then that turned into a permanent position. She's now also taking a MS degree in computer science at UCR online. She, of course, is still working at Illumina, but she began that, her job basically at Illumina from the outset of this program, well, at the outset of her internship. Last year, 4 of the 5 students have either accepted jobs or have already begun. One within a couple of weeks of graduating at a small company that is in Pomona, I believe. The other in San Diego. One in Irvine at IPS, and the other with me. So is that everybody? Yeah. So, I mean, it's a small sample size, but I'm actually quite proud of how quickly all of the individuals in the program have found, pretty gainful employment reasonably quickly, right?
That's pretty impressive.
Yeah.
Thank you.
I mean, I think that there's, of course, there's issues with the economy now because of our stay-at-home orders and COVID. But I don't anticipate that there'll be an enormous impact on industrial biotechnology. In fact, some of the key advancements that we need to get over this COVID issue will come from our industry, right? Vaccine development, vaccine production, testing is also intimately involved here, right? How do you manufacture the reagents for that? How do you design a new test for it? All those things are intimately involved in industrial biotechnology, and so. I can't predict what the economy is really going to look like, but I don't see a distinct lack of need for the products from this industry. Sorry, other questions?
Yeah, I have a question. Even though I'm in my 3rd year, could I still apply or show interest right now?
I don't think you apply right now. I think you apply next year because,
Okay.
of course, next year you'll still be continuing on and graduating as a chemical engineer or environmental engineer. But yes, I mean, what I would suggest for you is that you start to really look for research opportunity in one of the labs in our department or in a different department that's related to the center or to the program. So either myself or Dr. Good, Dr. Jinkerson, Dr. Lee, maybe Dr. Wyman's group, or you can look in bioengineering. There's a number of groups that would have kind of direct work related to this. If you can start kind of that undergraduate research trend now, that'll either roll right into a very obvious internship opportunity, or we'll give you some skills that you can now, make yourself a little bit more competitive for external internships, right? And so on the internship side, I can help identify opportunities, but I can't get them for you, right? So you still have to apply, and it's up to you to kind of secure that position. Certainly on campus, I can do a lot more than I can outside, but I certainly share all the contacts that I have. And we kind of curate a list of internship opportunities throughout the year for you guys to apply to. But so as a junior now, I would suggest that you look for those opportunities once we're back on campus and undergraduate research activities can resume. At this point, you probably know that on-campus research for undergraduates has been stopped until we move out of phase 2 reopening. Well, that's not to say that there isn't opportunities, because I have an undergraduate that works in my lab now, but she, of course, does it remotely. So,
Thank you.
yeah. Other questions? Yeah, please.
So for those who don't meet the automatic admission requirements,
Mhm.
myself included, what opportunities would you suggest we take, or how could we show that? How can we be competitive in applying?
I mean, our cutoff here will be about a 3.0 GPA, and so under that we have there are limitations from the grad division. And so there are minimum requirements that you have to meet from the campus as a campus standard. And those are 3.0 in your core field of study, I believe. But the specific details we could look up on the graduate division website. I think, participation in this group is helpful. I think strong letters of recommendation from your faculty that also helps. Research experience as an undergraduate helps. Of course, again, we get to the problem of that not being very accessible right now. But look, if you have a strong interest in coming to this program, you have a GPA of 3.0 or above, the chances are pretty high for you to be accepted into the program. So I'm happy to talk to you individually about that application, but especially for our own students, we know your background and where you've come from academically, and that's an easy acceptance for us to make.
Thank you for that.
There isn't too much I can do with a 2.5 GPA though. I mean, there's certain rules that we can't get around. 2.99 I can do something with, but. Other questions?
So when you're enrolled in the program, are you also doing research similar to the way you would when you're an undergrad and you're taking your courses, and after that you are able to participate in research? Is it something similar, or is that all put off till the internship period?
You can continue with your regular research opportunities throughout the year if you'd like. If you're doing that, you might want to roll those up into an internship opportunity. But I mean, our preference would be that you find an internship that you do in the summer at a company, right? And I think that's probably one of the strongest experiences you can have, right? That's an experience that I can't give you, right? We can't get you that experience in class. That's something that you need to go out in industry to get. If you're doing research on campus anyway, I think that there's enough time for you to fit that in, as long as you're a good scheduler, and that you're effective in keeping your time, right? If that's a significant commitment that you're making to that lab, and that work is related to industrial biotechnology, then there's certainly the opportunity to turn that into your internship as units, right? And so you might take those 298I units in the spring quarter. For example, if you've been, and this is what happened last year with a couple of students who were already completed the biochemical engineering option as an undergraduate, there was open time in their schedule where they just filled that in with 298I units. Instead of taking, say, for example, cell engineering or 124, because they already had taken those, they put those research units in there and completed their internships with Biolab. But we, had a dedicated project for them to do about, strain design for K. marxianus, actually. And so they learned some CRISPR genome editing tools and some other things. And so, of course, that's something that we have to talk about before you sign up for those, meaning we have to make sure that that research is actually fulfilling the role of being part of an internship for industrial biotechnology. But I see Ryan's on the call, and so maybe he can answer detailed questions that whether I was truthful or not. So Ryan is, of course, in the program now.
Yep.
Yeah, I'll take it. All right, I should log off because I have dinner duties soon. So, but Ryan, I will see you tomorrow at 1 o'clock.
Yeah. See you tomorrow.
Right. Great. Any last questions?
I think that's it. Thank you so much, Professor. I
Great.
really appreciate your time.
No problem. Yeah, no, thanks for having me. And I'm happy to do this again in another quarter, maybe in the spring, it's towards the end of the year when people are still for the people who don't know what to do yet with the end of their careers. We could do talk again. All right. Thanks a lot, guys.
Thank you.
Thank you.
Nice.